T4 bacteriophage, the LYTIC cycle. There are two kinds of cycles through which bacteriophages can replicate within their host cells, the LYTIC cycle and the lysogenic cycle. At the end of this video, you will be able to explain how the T4 bacteriophage through the LYTIC cycle multiplies within its host cell. T4 bacteriophage is a phage that infects E. coli bacteria. It contains an icosahedral head that contains double-stranded DNA genome. It has a hollow tail and there are tail fibers. These tail fibers help in attachment of the phage to the host cells. So the head is the part that has the genome, the nucleic acid, and the tail and related structures are involved in attachment of the phage, and these will help in the entry of the nucleic acid into the host cell. This phage, the T4 bacteriophage, is capable of undergoing only a lytic cycle and not the lysogenic cycle. So all these T phages, they are referred to as lytic phages because they always bring about the lysis and death of their host cell, Escherichia coli. The lytic cycle shows the following stages: attachment or adsorption, penetration, biosynthesis, assembly or maturation, and release. Let us have a look at each of these stages one by one. First, attachment or adsorption. The T4 phage comes in contact with E. coli. This is the surface of E. coli and initiates an infection. It recognizes the surface receptors on the cells by means of its tail fibers, and it attaches to the surface. It just comes and sits on the surface. This is known as landing. A recognition signal is sent through the tail fibers to the base plate. And now the short tail fibers will bind to the cell surface. The tail fibers of this bacteriophage are well adapted for this sort of thing, that is for this attachment, and this is known as pinning. And once it is pinned, the phage cannot move, and this is an irreversible process. The next process is penetration. So a few minutes after attachment to the host cell surface, this part where it is attached, this part of the host cell surface will get degraded. Now this happens because at the region of the base plate, the bacteriophage has a lysozyme. This is an antibacterial enzyme which will help in dissolving the cell wall at the region of contact. From where did it come? From where did this lysozyme come? Phage T4 packages a bit of this lysozyme in its tail base from a previous infection. So after pinning, the tail sheath contracts and it becomes shorter and thicker. And the tip of the tail opens, and through this hole now the double-stranded DNA will pass through this whole channel in the tail sheath and it will enter into the host cell. So basically this tail part is actually acting as a micro syringe, because as you saw in the previous slide, it's contracting and it is letting this DNA into the host cell. The capsid, it's remaining outside. It's only the DNA that passes into the host cell. We will understand the next step, biosynthesis, better with this illustration. Now, immediately after this viral DNA has been injected into the host cell, a process is initiated. This is the process for the synthesis of early proteins. Using enzymes and proteins that are available in the host cell, the relevant phage DNA gets transcribed into mRNA and translated into proteins. So these are the proteins which are needed to replicate the phage DNA. So when you're looking at the early proteins that are being produced, so among the early proteins, there is going to be a nuclease enzyme, that is a DNase, that will degrade the host DNA. So now once the host DNA is degraded, it cannot be transcribed. So only the phage DNA will be transcribed and translated. Another thing that is getting produced now is a virus-specific DNA polymerase. And this is also produced, and this will copy and replicate the phage DNA. Now, when the virus sat on this bacterial wall and made a small hole on the bacterial wall in order to inject its DNA, this hole is left behind, and that hole has to be plugged. So a protein will also be produced now, which is going to repair this hole. All these processes are possible only because the host enzymes continue to function. During this period, the cell's energy generating, protein synthesizing abilities are all maintained, but they have all been taken over by the virus. So at the end, what's happening now, you find that, look at this diagram here, what you find is that viral DNA is being produced in many copies. So a lot of phage DNA is getting produced. So the next process is late proteins have to be synthesized. So that's the next step. Now that the phage DNA is produced, it can be used for transcription and translation to make the next set of proteins. And those are the proteins which are known as the late proteins. The late proteins are mainly structural proteins, and these are the proteins that will make the capsomere. These are the proteins which will make the tail and tail fiber. Another late protein is lysozyme, and this will be packaged in the tail of the phage, and this will be used later while the phage is escaping from the host cell. So basically, during the process of biosynthesis, the three important processes taking place are degradation of bacterial chromosome, replication of phage DNA, and synthesis of phage proteins. Now that viral proteins and viral DNA have been replicated multiple times, they all need to be assembled together. This is known as assembly. And the whole process of assembling once the phage particles are all assembling, we call it maturation. We are saying that the phage particle maturation process is taking place. The proteins that make up the capsomeres, they all get assembled and they will take in the DNA. They reel in a copy of the phage DNA. Then the tail and all the associated structures, the sheath, the base plate, all these also assemble. And once the head assembles, then the tail gets added to it. And while the viruses are assembling, as a late protein already mentioned in the last slide, lysozyme that is being produced. Part of this lysozyme is just stored in the tail, and the rest of it is going to be used to break through the peptidoglycan cell wall of the host. And that will accomplish the lysis of the host cell and the release of the mature viruses. So once the viruses are released, they will go to the nearby cells and they will infect them too. So this whole period from the entry of a virus into a host cell and the appearance of infectious progeny, this is called as the eclipse period or the latent period. This duration is around 25 to 35 minutes. And the burst size means how many viruses are released per cell. And this, the number is around 200. So each time a single bacteriophage enters into an E. coli, at the end of all this process, from one phage, you get about 200 phages now being released from the host cell. And because the host cells are killed by lysis, this type of infection is also referred to as a lytic infection. So as you can see here, whatever is released from the host cell is going to continue the cycle by infecting other bacterial cells. The lytic cycle is, of course, only one of the two cycles of viral reproduction, the other being the lysogenic cycle. The lytic cycle is typically considered the main method of viral replication, and it results in the destruction of the infected cell. What happens in the lysogenic cycle, and how is it different from the lytic cycle? We will be looking at this in another video. Thank you.